Thank you, Toda Rabah. It's really a pleasure to be here. And I think, first of all, again, it's an honor to be here again. I haven't been to Israel in about 5.5 years since I was here and kind of gave a bunch of lectures a number of years ago. I met several of you at the time. And I'm really honored to have Dr. Abraham Niska here. I don't know how many of you know him, but Abe has been what we call an NIEHSer, which means that he's been really part of our institute for many years now. For many years, he was actually at the institute. And for the last 10 or 12 or so, he comes about once a week every 6 months and helps us make sure that our pathology is completely up to stuff. So I was really honored to find that Abe was going to be able to join us here today. So what I really want to briefly talk about is some of the cutting-edge research that's going on in environmental health. And what I want to do is kind of tell you about what's happening in environmental health. While certainly in many parts of the world, and certainly in the developing world, and even in a country like Israel and in parts of the United States, we still have some interest in acute toxicology. What does a really high dose of a chemical do? You know, are we getting overt toxicity? Are we actually, leading to someone losing consciousness, or are we actually killing someone? And the old paradigm was that we deal with chemicals that act by overwhelming the body's defenses at very high concentrations. The new paradigm, where we're really beginning to spend much of our effort today, is understanding that low doses of chemicals can have very major effects. Because in fact, many chemicals actually act like hormones. And we know that hormones act at exquisitely low doses, for example, often in the picomolar range, and can alter basic physiology. And that the levels of exposure to which that occur in the environment to some of these chemicals are in the range of hormonal substances. And in fact, we're beginning to find that they do have effects. The other really, I think, paradigm shift that we have is that we used to look for exposure to a compound and you'd get exposed and immediately you'd see an effect. Or maybe the next day you'd see an effect. What we're understanding now is that because that chemicals can have effects which you don't know about those effects for many, many long years. And part of this is the whole science of epigenetics. And I'm going to talk about epigenetics a little bit more and what the mechanism means here. But I should stress that much of the work that we do and that we sponsor at the National Institute of Environmental Health Sciences is basic mechanistic biological research. Because if you understand how a chemical causes something, not only you can prevent it, but it gives you better predictive power in understanding why another chemical might do something as well. So again, some of the new environmental health paradigm is that we have often looked at exposure to adults. And in fact, and I think Abe Niska could tell you, we often, when we do our testing paradigms, take we take adult rats or adult mice, and we expose them for 2 years for chemicals, and then we see what has happened. And I would argue that we've missed the most sensitive and the most susceptible period in development, which is the in utero period or during the infantile period. And in fact, that's when events can actually occur. So we're beginning to understand that early life exposure can alter your susceptibility to disease for the rest of your life. Again, as I said briefly before, is the susceptibility to disease can last long after this exposure is gone, even decades later. So that if you do something during development, during a critical window, that change in how the organism will respond is with you forever, which may mean that you respond differently to a secondary insult later on. The third really major new point that these chemically induced responses are most likely related to changes in gene expression or changes in the regulation of proteins. They're functional changes. We're not looking as much today at gross structural changes that can occur at high doses of chemicals. We're looking at how early life events can change how we respond later on. And these functional changes are associated with altered programming, and they often permanently alter the gland or the system potential. So I'm going to give you some examples later, or I'll mention one now. We know that early life exposure, for example, can alter, and this can occur in rodents in utero, and we're not sure whether in humans it's occurring in utero or during infantile period, but you can totally alter the growth, for example, of the prostate gland, or the mammary gland. And so that later on, at puberty, that gland doesn't function appropriately. Nothing happened to puberty, it's what happened way early on. So I think it's important when we talk about environmental health to realize that the environment can be talked about with a capital E, at least in English for environment, meaning it's not only chemical exposure. It's also issues like stress, nutrition, drugs, and infections. And in fact, we're beginning to understand that, for example, your susceptibility to an infectious agent may be altered by what kind of chemical exposures you have. And that includes stress as a chemical exposure. We all know what is stress really. You're changing your whole corticosteroid level, and that alters how you respond to things. But for example, there's some recent studies that have come out, especially studies coming from Denmark and the Faroe Islands, showing that children who were exposed in utero or during lactation to very low levels of chemicals like PCBs, in fact, have difficulty mounting the normal vaccination response. So if you try to vaccinate them against pertussis or whooping cough or a number of other diseases, they don't get a robust response. Their sensitivity has been altered. So again, the new disease paradigm is that the environment leads to changes in gene expression, which may be due to epigenetics, and I'm going to come back to that, permanently altering the glands or the organs, and that persists for a long time and increased susceptibility. So what is epigenetics? Well, first of all, we might call the last decade or the last 2 decades the decade of the genome. We all know that it only took 10 years instead of 20, and it cost about $3 billion to sequence the first human genome. Right now, you can sequence an entire human genome in the matter of a week for about $10,000. We expect that within the next couple of years, anybody really who wants to have their genome sequenced for whatever you want it for, can do it it will be done in probably a matter of days and be down to about $1,000 per genome. But what we're beginning to understand is, while that's been the exciting new scientific findings of the past decade, I look at the next decade as the decade of the epigenome. And what the epigenome is, those are all those changes that occur to the packaging of DNA in your chromatin, in your cell, that alters how DNA is expressed. When you have epigenetic changes, there is no change of the basic A, G, C, T structure. You still have those same bases. You're not having a mutation, either a point mutation or a deletion or an addition mutation, same A, G, C, T, but you are changing how it gets expressed. And by the DNA, what you're doing is you are methylating, especially cytosine residues, in the DNA sequence. And when you tend to methylate these Cs, you tend to shut off gene expression. When you demethylate the cytosines, you tend to turn on gene expression. But it's not just changes in the cytosines of DNA, and some work that we sponsored, a paper in Nature, showed that last year you could even methylate adenosine residues. Well, that is a whole new paradigm. Also, within the past couple of months, we're showing that not only do you methylate some cytosines, but you can get hydroxylation of these methylated groups on cytosine. And again, that has kind of a contrasting role. Methylation of the Cs tends to shut off the genes. If it gets hydroxylated, it tends to turn it on again. So you have different levels of control of the production of RNA from the DNA. But that's not the only type of change that epigenetics involve. I think you all know that the DNA is not freely floating around in your nucleus. It, in fact, is rolled up on proteins called histones. There are at least 4 kinds of proteins. And these proteins are composed, obviously, of strings of amino acids. And many of the amino acids have residues, such as lysine, which can themselves be methylated, or acetylated, or propionolated, or butyrylated, and doesn't only have to be methylated, it can be dimethylated or trimethylated, and I could continue. And what you have is a great amount of combinatorial complexity of how you can modify these histones, which affect how the DNA gets packaged into the chromatin. So now you have alteration or modification of the cytosines and maybe the adenosines in DNA. You have modifications of the histones, and that all controls how your chromosomes are packaged in the cell and how they get turned on and turned off. Now, during development, you have waves of epigenetic changes that occur. So that, for example, in the mouse, at about gestation day 13 out of a total of 18 days of gestation, you get a whole wave of methylation that occurs in what will develop into the reproductive tract. And on day 17, just the day before parturition in the mice, you have a wave of demethylation that occurs. If you change these patterns that occur, that in other words, if you delay the epigenetic programming, or if you accelerate it, you totally mess up the appropriate structures. Now the beauty, and this is maybe backwards, epigenetic programming, is it's not a mutation. And there are at least opportunities to reverse the specific methylation. So many of you may know that a lot of the anti-cancer agents, some of the drugs that are used in treatment, for example, of HIV/AIDS, in fact, are epigenetic agents. They are altering the methylation, especially of DNA. Some of them are altering the methylation of histone, for example. So that there may be opportunities here to reverse changes that occur. The other point about epigenetics is we all know that you inherit your genes from your parents and you pass them on to your children. There is now evidence that your epigenetic profiles can also be inherited. And it's clear that they can be passed on from parent to child. In animals, there's evidence that these can be passed on for as long as 5 generations. In humans, there's some evidence that maybe we can pass this on as to grandchildren as well. But this is all very new, very exciting information that we're just beginning to understand. But again, while genetics involves this primary sequence of AGCT in your DNA, epigenetics involves modifications of the bases and the proteins that alters how your genes get turned on and turned off. And that's where a lot of the exciting understanding of how environmental exposures can affect how we grow, how we develop, how we function. So epigenetic changes have now been implicated in a wide variety of human diseases. As I mentioned, there are normal waves of epigenetic programming that occur during parts of development. We're also finding that epigenetics is playing a key role in the processes of aging. So again, maybe some opportunities for intervention here. There are many ways that you can alter the epigenome. As I mentioned, environmental exposures is one of them, but that includes nutrition. You know, somehow we forget that when you eat, what are your nutrients? Your nutrients are just kinds of chemicals, right? Just like your drugs that you take, pharmaceuticals, are kinds of chemicals. And all of these things can alter our epigenomes. We know, for example, that heavy metals, for example, have epigenetic influence as well. We know that drugs of abuse can have epigenetic changes as a result. And we know that actual infectious agents, not only HIV, but for example, H1N1 influenza, which was a pandemic across the world last year, can lead to epigenetic changes. And there are a number of diseases that now we know there's evidence that these are related or can be caused by changes in this expression of DNA. Cancer is obviously one of them. In fact, that's the one we have the most information about. But also heart disease, autoimmunity, obesity, diabetes, neurodevelopmental effects, and so on. And one thing you might notice when you look at some of these adverse health outcomes that I have listed here, certain of these things are in epidemic proportion today, certainly in my country. I know that some of them are increasing in Israel as well, but there's a great increase in obesity, a tremendous increase in type 2 diabetes, tremendous increases in various types of autoimmune diseases. We all know that certain neurodevelopmental disorders are increasing. In our country now, the incidence of children with autism is now 1 in 110 children. And the ratio of boys to girls is 4 to 1. And I believe that autism is also increasing dramatically, I've heard, in Israel as well. We know, for example, that ADHD, attention deficit hyperactivity disorder, is increasing dramatically in our country, as well as other forms of learning disability. Many of these things are occurring, or the incidence is increasing too rapidly to be explained by genetic changes. Our genes don't change that fast, but our epigenomes can. So I was just going to point out, before I'm going to come back to epigenetics, Alyssa, but I wanted to point out some of the really new data that is showing associations between some of the diseases that are increasing rapidly in the population and exposure to environmental agents. So this is the agricultural health study is a study that's been going on in the United States between my institute and the National Cancer Institute and the Environmental Protection Agency. And we started this study in 1994. And we're looking at almost 32,000 licensed pesticide applicators in the United States. We're also looking at their spouses as well. And we've been, as I said, the study started now 17 years ago. And we've been following them repeatedly at different intervals. And what you can see here, the red line is the base of an odds ratio of 1. So that's where we would expect the level of type 2 diabetes to be. And the black lines are showing you, in fact, what the increases are. And you can see that for a number of pesticides, several organochlorines, several organophosphates, and then even something like chlorotrazine, and I understand that there's a lot of talk about atrazine in Israel, and that may actually have recently been proposed to be banned or something. Atrazine and chlorotrazine are in the same family. What you can see in almost all these cases, there are significant or near significant increases in diabetes associated with the use of some of these pesticides. So we also, we held a meeting in North Carolina about a month ago where we were trying to do some kind of meta-analyses, and we were looking at the role of environmental chemicals, not only in type 2 diabetes, but also in obesity as well. And what we found is that for a number of the persistent pesticides and some of the persistent chemicals like the PCBs, there appears, if you look at all the studies that are out there, and these were both prospective and cross-sectional epidemiology studies, and these were the studies that were the most robust. And a few questions later, I can go into more detail about the specific choices of the studies. But you can see trans-nonachlor is the major metabolite of chlordane. Chlordane obviously was one of the Stockholm POPs, but it's still out there. We know that many of these chemicals are so persistent they never go away. You look at DDE, and in the great studies, especially the prospective longitudinal studies, what you can see is significantly increased risk of diabetes associated with exposure to these chemicals. And then, for example, if we look at both the prospective and the cross-sectional studies for PCBs, again, what you can see, in general, there appears to be an increase in association with diabetes. So that there is evidence now that there are pesticides that are associated with increase in type 2 diabetes. Now, I should not eliminate type 1 diabetes is also increasing in incidence quite dramatically. Again, at least in the United States, type 1 diabetes is basically an autoimmune disease. And as I said, autoimmune diseases appear to be increasing. There isn't enough information for us to pull together yet that association, for example, to see whether that's association with pesticides. So I mentioned before that in some of the paradigm shift, we're talking again, is that there may be times in our life when we are more susceptible than other times to exposure, whether it's to environmental chemicals or to drugs or to stress hormones or to infectious agents, but development is one of them. And why is development in a susceptible window? And part of that is it's a time of rapid growth and active cell differentiation, very rapid metabolism. The immune system is developing during, for example, the infantile period and some of the in utero period. There are a lot of opportunities for initiation of lesions and promotion of altered cells. Development is highly integrated. I use the example of epigenetic programming and of how if you shift the timing of that, you have major effects that can occur. And again, it's during development that many of these epigenetic marks are actually set. I should mention it's not only in utero and infantile that's important, it's also adolescence as a sensitive time. And many of you are probably familiar with this basic paradigm that there are different windows that occur during development. So if we're looking at heart development, the heart is most sensitive during that first trimester of human development, actually during the first 8 weeks. If you're looking at the eye, most of that is in the very first trimester. If you're looking at something, though, like the central nervous system, that has multiple times where it's susceptible, so that not only is it susceptible during the first semester, the second trimester, and the third trimester, but we now know, and anybody who's, well, we've all been teenagers, but who has a teenager at home know that is not a stable period emotionally. And we know that actually that the neuronal growth, the neuronal synapses are still forming up until about 25 years of age. So again, different times may have different windows of susceptibility. And it's also important that, for example, I think one of the best examples that we know from the human literature is if you look, for example, at retinoids. Many of you know that Accutane is a synthetic retinoid that is used for the treatment of severe acne. And you know that very early in development it will affect, for example, eye growth in the heart. Later in development, if it's given it will affect the limb development. Later on, it can have other effects. So again, when you have exposure to certain chemicals is going to affect not only the organ system, but exactly what part of an organ system might be impacted. So I think that's just something we need to remember. And then this is the whole, there's a whole new field, there's even a new journal and a new society called the Developmental Origins of Disease. Sometimes people refer to this as FEBAD, the fetal basis of adult disease, but it's really is that many times during our life we have windows of susceptibility that can lead to effects that are seen later on. So I really should have a lot more arrows here because, for example, it's not only effects that can occur during gestation that can affect you during childhood or puberty or your reproductive life or middle age or later life, but in fact, there are times in puberty that can affect your reproductive life and later on. And there are even, for example, times in your reproductive life. A pregnant woman, not only is she inherently susceptible because of her developing fetus, but the woman herself is at an inherently susceptible window. Think of her breast development in order that she'll be able to lactate. And so you have basically a time when changes will be noticed later on. So that we have this whole developmental stressors at multiple times that can lead to effects that you will be noticed or will be functional later on in life. So talking about windows of susceptibility, and I was actually talking to Ruth Ostrin while we were driving here, this is some evidence that we've recently pulled together about maternal smoking and obesity in children. I think we've all known for a long time that maternal smoking is not a good thing for the developing fetus. Children are often low birth weight if their mothers smoke. We know that if a woman smokes, her child is much more likely to have asthma than if she didn't smoke, although that is actually a very interesting gene-environment interaction because there's a specific polymorphism in one of the glutathione transferase genes, and if the infant has that specific polymorphism, and if the mother smoked, that child will have asthma. And if the infant doesn't have that polymorphism, even if the mother smoked, the child probably won't have asthma. So there's gene-environment interactions being very important. But this is some recent data, again, that we've been pulling together. And what this shows you is the red line, again, is just showing you the, risk of, the basic risk of 1. And what you can see, whether you look here at overweight or obesity, or the blue lines are type diabetes, children whose mother smoked when they were in utero are much more likely to be overweight and or obese and diabetic, type 2 diabetes. And if there are any endocrinologists in the room, I know that I have a very good friend who's a pediatric endocrinologist, and she tells me when she was in training 25 or 30 years ago, she never saw a child with type 2 diabetes. And now 90% of her patient population are children with type 2 diabetes. So I think this is a pretty important understanding that the odds ratio for obesity is at least a 1.5 and for overweight is at least 1.6. I mean, I think this is a very telling piece of information. Now, I in no way want to take away the responsibility of people for controlling their weight by exercise and diet, but in fact, if prenatal exposures are setting you up to be more susceptible that's something that we need to know about and potentially work on doing something about. So I mentioned before some of the paradigm shift that we're beginning to be more concerned about low-dose exposures as opposed to the high acute exposures. And I mentioned that our hormones often work on picomolar amounts of compounds that profoundly control our basic physiology and have profound effects on development and normal health. But what we're finding is that very low-dose chemical exposures can also affect our endocrine systems, and what's important is you have different things that can happen at different doses. So, for example, a very low dose of a chemical may perturb a hormone system, while the high dose of the chemical may actually cause overt toxicity. Some of the early work that's been done with the chemical BPA, bisphenol A, I'm sure you've all heard about it. That's the thing that used to at least be and may still be here in Israel in hard plastic baby bottles. It's also in the lining of a lot of cans. It's actually present in thermal paper that you get from cash register receipts. We're finding that BPA at high doses, for example, can cause actual, for example, in mice, affect the growth of prostate gland, and at very high doses it may actually be toxic to the prostate gland. You get a very small prostate gland. At very, very tiny doses, at doses that are in the physiological range, in fact what you find is the prostate gland undergoes a hyperplastic response and gets too large. That has long-term consequences for the mouse offspring. But we find for a number of chemicals is that you don't have the straight, what we think of linear dose response curves. The old paradigm is the dose makes the poison, but now we understand it's not just the dose, it's the dose plus the timing, plus the inherent susceptibility that makes the poison, and that you get different effects occurring at different doses. And it's important to understand our animal studies, our toxicology studies, are often people will say, oh, they're so high dose that they're in fact irrelevant to human exposure. But you usually have to give somewhere between 20 and 100 times the same administered dose, the same concentration in the water or concentration in the air to a little rat or mouse that you do to get the same internal concentration that you do in humans. And that's a point that's often missed. So that you have to use a higher level to expose your animals to get the same blood level as to have it comparable to a human study, for example. An example I have up here is that very low doses of BPA change brain structure and function and behavior. And we now have at least 2 studies that have come out of the epidemiology literature showing that depending upon the mother's first trimester exposure to BPA can lead to altered behavior in her 2-year-old offspring. We're also looking at a wide range of health effects. Endocrine systems really control our normal physiology and govern every process in our bodies. And we understand that when chemicals act on our hormone systems, we can see these effects in multiple organ systems and under different conditions. So that we're seeing that when you perturb endocrine function, we're not just looking at effects on the reproductive system. And I understand that you had a wonderful workshop here a couple years ago looking at endocrine disruption and reproductive function. But our endocrine organs our endocrine hormones, I should say, interact very, very closely with our immune system, with our basic metabolism, with our neurological system. Everything is integrated. And I think we've known for quite a while, really, if you affect one hormone system, you also affect other hormone systems because our bodies are in pretty delicate balance. So again, I've already talked about the weight gain issue later in life. We know that from our animal studies, and I showed you the data from human studies showing that prenatal exposure to tobacco smoke is associated with obesity, not so much later, actually in the children at age 10. But again, endocrine disrupting chemicals have been linked to cancers, behavior, diabetes, immune function, reproductive function, and heart function. And another point, which I made before and I'm coming back to, and I'm kind of hitting you over the head with it, is that these health effects of exposure, things that perturb our hormone systems, can be observed long after exposure has actually stopped. So I sometimes use the expression of hit and run. You have that effect early on during a critical window of susceptibility, and it leads to a permanent change that we see later on. Again, many of these changes we're now understanding are via changes to epigenetics. Our genes are permanently altered in how they're turned on and off. And we are busy conducting many studies on these long-term effects of endocrine exposure, looking at studies of children with behavioral, physical abnormalities, for example, who were exposed not only to something like BPA or pesticides or PAHs, but to chemicals like phthalates, which are present in many plastics. And there are recent studies that have been shown coming out of Columbia that if the mother has elevated, well, at the high end of the normal range, really, of phthalates that can be shown during pregnancy, her children are likely to have lower IQ. Also, their increase in ADHD, the attention deficit hyperactivity disorder. Some studies with PBDEs, which are flame retardants, and I'm not sure what the requirements were in this country, but in my country for many years, in order to provide fire safety, companies were putting high levels of brominated flame retardants into many of their, for example, furniture products as well as their electronic products. And what we're finding is that developmental exposure to PBDEs are associated with cognitive deficits in the children. And in women, we're finding that long-term exposure to these compounds are associated with decreased fertility as well. Now, when I talk about the cognitive deficits, what I should mention is that when we're dealing with environmental chemicals, in general, we're not talking about, as I've said before, acute poisonings. We're talking about low-level effects. So you can't look at a child, just like with lead, which I understand was just removed from your gasoline last year. But you can't look at a child and say, okay, Chaim, if you had had, a little less blood lead as a child, you'd have a couple more IQ points. Okay, but you can look at a population of children and see that that distribution is shifted to a lower IQ. It's the same thing with a lot of these environmental endocrine disruptor compounds. We've seen it with PCBs. We're seeing it, for example, with the PBDEs. We're seeing it with some of the heavy metals. Again, shifts in the distribution, putting more children in an at-risk population. So again, cancer and birth defects are not the only outcomes that we're concerned with from environmental exposures anymore. I think a point that needs to be stressed is that what we're focusing on now, especially in the developing and the developed world, are complex diseases. And just like most complex diseases are going to have complex genetics, they're also going to have complex environmental causes of these diseases. There's going to be very little one chemical, one response, or just like there are very few diseases today that are based upon a single Mendelian genetic cause. So the diseases like obesity, diabetes, heart disease, cancer, autoimmunity, etc., are diseases where the environment is acting through epigenetic mechanisms. And complex diseases, complex causes, nobody is exposed to one chemical at a time. We are always dealing with mixtures of chemicals, and we're exposed to many. And the question is how do we deal with this, especially when we get into talking about a regulatory agenda? How do we deal with the fact, if you're an occupational worker, if you're working in a factory, and your factory is making vinyl chloride, and we all know today that vinyl chloride causes liver cancer as well as some other cancers as well. You may be dealing with one chemical in excess. But when we're dealing with the general population, we're all exposed to many chemicals at a time. We all have exposures to certain pesticides. We're all exposed to BPA and phthalates and flame retardants. And the persistent chemicals that were used 20 and 30 years ago and are no longer used, but they're still in our bodies. We're all exposed to certain metals. We have this mixture. How do we deal with it? Well, one approach that's been used successfully for a class of chemicals, the dioxin-like chemicals, I think you've all heard of TCDD, which is often called the most toxic man-made chemical, but it's just the prototype for a class of chemicals. And we can treat all those chemicals as if they were a dilution of TCDD, and we use a weighted potency scheme. We basically add up all of these different chemicals, and we can use bioassays to determine how much dioxin-like activity do we have in a mixture. So some of you may be familiar with there's been a recent dioxin poisoning in Germany. where they found that some of the food has been contaminated with dioxins. Something similar happened in Belgium in 1999. It was very sad because the chocolate was contaminated, so there was no Belgian chocolate for a while. But anyway, the point is that we can deal with that class of chemicals. A similar approach is being used now for the organophosphates, based upon cholinesterase inhibition. And you can, again, kind of add up all the activity of all the cholinesterases and treat it as single exposure. Excuse me. But again, part of that, the importance is that we're not just exposed to dioxins, we're not just exposed to OPs, we've got lots of other classes of chemicals that we're exposed to. So how do we begin to deal with this multiplicity of exposures? And the answer is we don't really know. We're finding that if you're looking at a certain kind of effect, say you're looking at male reproductive effects, we're finding that you can in fact add up the chemicals in a kind of response addition kind of mode. So if you deal with something that says blocks testosterone synthesis, sorry guys, losing my voice. Something that blocks testosterone synthesis, something that binds to the androgen receptor. Thank you very much, Maya. So that it, that if it binds to the androgen receptor, you can't have testosterone action. Maybe something that is involved in testosterone breakdown. A number of different points in the action of male hormones. You can actually add those up in a response addition mode and predict what would be the effect. But again, we're not just being exposed to things that affect our male hormones. We're being exposed to things that affect multiple systems. And how do we approach that? And I'm going to give you one idea in a moment. But that it's just something to put back in your mind. We've got to begin to understand how do we deal with the multiplicity of exposures that we had. Another issue is routes of exposure. For certain kinds of chemicals, the route of exposure is going to be very important for the effect. So if you're dealing with ozone, ozone really never gets much beyond your respiratory tract. So you're dealing with a point of contact toxin. But for many chemicals, it doesn't matter how you're exposed. Once it gets in your body, you have systemic toxicity. So we may need to understand that depending upon the kinds of chemicals or the kinds of exposure we're dealing with. The example that I'm pointing up here is hexavalent chromium. I don't know if that's a problem in water supplies in Israel or not. No? Well, you're lucky. It is in the United States. In about 26 states, we're finding that hexavalent chromium has contaminated the water supply. Chromium is a naturally occurring element, obviously, but usually in the environment it's in the trivalent state, which is not very toxic. But it's used chemically, for example, in electroplating and a lot of different other industrial processes. And if it gets into we know that if you inhale it, we've known for years that hexavalent chromium is a respiratory carcinogen. Our institute, we're responsible for listing chemicals for the United States government as known or reasonably anticipated to be a human carcinogen, and we listed hexavalent chromium over 30 years ago as a known human carcinogen if you inhaled it. But industry has tried to say for a long time that it's not a carcinogen if you drink it in water. So the National Toxicology Program actually did a study. Abe, I don't know if you actually read those slides or not, but we exposed rats and mice for 2 years to hexavalent chromium in drinking water, and the concentration that actually gets into the animal is only maybe 10 times the concentration that some people have been exposed to. Excuse my voice. But anyhow, what we found was that the animals all ended up with tumors, both the rats and the mice. The mice had intestinal tumors, and the rats had especially tumors of their tongue and oral cavity. So either way, hexavalent chromium, either inhaled or oral, is a problem. And we're beginning to find that, people have known for years that arsenic, and I'm going to come back to arsenic, can't remember if that's now or later today, as a carcinogen, but we know that arsenic inhaled is clearly a lung carcinogen. We know that arsenic ingested is a multi-site carcinogen. Okay. Oh, here's the arsenic right here. So the point here is that we've been doing a lot of studies on arsenic because Arsenic is one of those really examples where the World Health Organization tried to do something really good. And in the 1960s, there was tremendous surface water contamination with microbes. And people were ending up with really bad gastrointestinal diseases. And in India and Bangladesh, they sunk wells into the aquifers that were about 25 to 30 meters below the surface. And the water that came up was clean and pure, and there was no microbial contamination, and the people loved it. But 10 years later, they started finding people developing Blackfoot disease, which are severe hyperkeratosis and hyperplasia and pigment in their extremities. And people actually went on to develop severe arsenic poisoning in these areas. It took a while before people started analyzing the water and found out that the concentrations of arsenic in the water were, instead of in the range of 10 to 50 parts per billion, they were in the range of some as high as 10 parts per million or higher, phenomenally high concentrations of arsenic. And this mistake, which was done with all the best intentions, has been repeated not only in India and Bangladesh, but in Vietnam, in Inner Mongolia, in several other parts of the world. And in many places of the world, in my country, in the Southwest, and in parts of the Northeast, in New England states, in Maine and New Hampshire, you have naturally occurring arsenic getting into the water at concentrations that can cause problems. And so we've known for a long time that arsenic can cause bladder cancer and liver cancer and kidney cancer, but now we're finding that arsenic, drinking water with elevated arsenic, is associated with cardiovascular effects, with diabetes in adults, and with developmental effects. And unfortunately, because of this poisonings, the unintentional poisonings that occurred, we know when it started, and we know when the problem was discovered and it was stopped. So we actually have people that we're following who were exposed in utero and maybe in their early childhood, but not when they became adults. And we have people, for example, that were only exposed after, say, in their childhood and adults. We have different populations. And we're able to show, for example, that elevated arsenic in water during gestation is associated in the offspring with lower IQ. And with, by the time they're 20, these people have severe lung effects, and we're beginning to see some cancer as well. This is just an example over here of some of the severe skin lesions that can occur, as well as skin cancer in populations that are associated with arsenic. So what I should point out is some of the effects that we're seeing with arsenic are not all the high-dose effects like the Blackfoot disease and the skin cancer. We're finding that these associations with some of the cardiovascular diseases, some of the neurologic diseases, and some of the cognitive effects are within what used to be the regulatory limit in the United States, which until 2006 was 50 parts per billion. We've now lowered it to 10, which is the World Health Organization standard, but there's some suggestion that that may even not be protective as well. So how do we deal with all these complex chemicals that we're dealing with in all of them? I think we're beginning to develop some predictive toxicology approaches. So I just wanted to tell you a little about the new screening approach we're using. If you're doing, for example, epidemiology studies, we said 1 to 3 studies a year. If anyone, some of you are doing environmental epidemiology, you're often looking at projects that are going on for 5 or 10 years or even more. The most powerful epidemiology studies are your prospective longitudinal studies, and if you're looking for effects that take a long time to occur, you could easily be waiting 5, 10, 20 years or more to see your effects. Our standard toxicology studies, where we might dose rats and mice for 2 years, males and females for 2 years each. I should mention that all of our rat studies, we're not starting now at adults, we're starting now in utero with exposures so that we get that most susceptible period, but then holding the animals till they're 2 years old as well. There's not enough money in the world to do more than somewhere between 10 and 100 of these per year. It's extremely expensive, take a very long time. From the time we conceptualize one of these 2-year rodent studies to the time we actually report out the data, we're looking at a minimum of about a 6-year time period. Sometimes it's a little bit more if the pathology turns out to be complicated and complex. Well, we're beginning to develop some alternative animal models that we can use. There was an article in Nature this week about using Daphne because its genome was just totally sequenced this year. And that's used, but we have a lot of studies that are, people do studies with Drosophila, people do studies with C. elegans, the worm. People do studies, for example, with zebrafish, which have the beautiful property of being totally transparent during the embryonic stage, and you can actually watch the organs developing. And they have a relatively short lifespan of about 3 months, so that you can get a great deal of information, and they're only about this big, so that you can also have a large number of animals. Of course, many people say that animals aren't, these animals aren't human beings. But in general, if we find that we perturb basic physiological process in different animal species, and we see it in more than one animal species, it's likely that at least some people are going to be susceptible as well. But in order to deal with the tens of thousands of chemicals in commerce that exist, and to do large numbers, we need to start going to some of the rapid in vitro screening assays. So we have a program called Tox21, which is dealing with kind of a shorthand for toxicology in the 21st century, where we have developed screening assays where we use not hundreds, but thousands of chemicals, can be screened in a short period of time, not by one cell-based assay or 10 cell-based assays, but by hundreds of cell-based assays. So we're about to do a study where we're going to be running 10,000 environmental chemicals through 100 different cell-based assays in less than a year. We've already started this with smaller numbers. We've run about 400 chemicals through about 400 different assays, and I'm going to show you a little of that data. These are collaborative studies that are being done through my institute, the National Toxicology Program, through the National Human Genome Research Institute, which is another part of NIH, through the US EPA, and through the US Food and Drug Administration. So it's kind of a nice example that doesn't always happen in the US, of different federal agencies working together. But what we're going to get from this is not only the information that this chemical does X, affects this cell type. We're actually going to be looking at critical toxicity pathways. This will all be analyzed. We're generating huge amounts of data. Nothing that any one person can do requires tremendous bioinformatics capability. And I don't know what the situation here in Israel, but when I look at the big shortage of scientists that we have right now, we really need more bioinformaticians who have the ability to work with huge amounts of data that we're getting, not only from the deep sequencing of the genome, but the deep sequencing of the epigenome that's starting, as well as all the rapid screening. And we believe this is going to give us some predictive power to understand, for example, what new chemicals and what mixtures are doing. So I just wanted to give you an example of some of the data that's coming out of this. And this is through, again, some of the first studies coming out of here through the ToxCast part. And what we have here is we tested 309 different chemicals. Many of them were pesticides, for which we had a great deal of animal data, in some cases even human data, as well as in vitro data. And then we put in this, for example, something like BPA, bisphenol A, which is this chemical that is now in commerce, and that, at least in the United States, the recent studies show that at least 94% of the population have measurable levels. And the analysis is something that we do is something called ToxPi. So you end up with a pie where we're looking at different kinds of assays. And we were really looking for the endocrine disrupting or the endocrine active action of these chemicals here. So we looked, for example, up at 5 different assays. The TR is thyroid receptor assays, and ER is 5 that deal with estrogen receptors, and AR is 5 that deal with androgen receptors. We looked at the ability to interact with a bunch of other nuclear receptor genes, not only things like glucocorticoid receptors, but receptors like PPAR gamma, which is often related to diabetes, for example, and many of the other receptors. We looked at physicochemical properties, like, what's their partitioning ability and their optimal water partition. And we looked at a number of different pathways. And all those colors, they're all put together, and you see how many of the different assays that are run that you have activity. And I use a term, instead of all the specifics, I say, what really lights up the board? where do we see a lot of effects? And what you can see is the number one most active chemical in these 309 chemicals that were looked at is HPTE. HPTE is the estrogenic metabolite of the pesticide methoxychlor. And we've known for years that that's highly estrogenic. That's number one. Number two was BPA. Okay? And this is, some people have questioned whether BPA is really an active chemical. Well, running it through 400 different assays, it's the number two chemical. Methoxychlor, which is the parent, has to be metabolized to HPTE, is still a pretty active chemical. Interesting to me is that this is all these little blue dots, those are the 400 chemicals that were assayed, and this is giving their activity here. Rotenone, which is a pesticide that's used, and recent studies from the Ag Health study have shown that that's associated with Parkinson's disease. And that's got a fairly good amount of activity. One chemical I don't have up here, which is right up there around methoxychlor, is triclosan. Triclosan is found now in drinking water a lot, but it's the antimicrobial agent that's present in a lot of your hand sanitizers and a lot of your antimicrobial soaps and a lot of toothpaste. And there's been some concern because that's been shown to affect thyroid homeostasis, and recent studies have shown that it impairs estrogenic activity as well. So we think that we're finding some of these analyses very exciting, certainly very stimulating to think about. We've got a lot more chemicals to look at, but as I say, within the next year we're going to be testing 10,000 chemicals, and I think we'll really be able to begin to understand which ones are active in different kinds of pathways, different kinds of predictions, different kinds of receptor activities, and give us a better handle of how to approach the vast number of chemicals, but also the potential here for dealing with mixtures. I can tell you this is done with huge robots. I don't have a picture of the robots here, but there are robots that are, they're 1536 well plates. So you can test, even with all the controls you put on a plate, we do 15 dilutions of each chemical on these plates, so you have very detailed dose response curves, you have estimates of potency, but again, we can run through over 10,000 chemicals a year in these studies. And right now they've already been doing this, looking at different potential structures for different pharmaceutical agents that are being developed as well. So, another point I wanted to make is, where are we going? Well, we've got to deal, if you're dealing with environmental, you want to deal with some of the emerging issues. Not only the chemicals we already know are out there, but what about the new chemicals? And I'm sure that Israel is probably a center for the nanotechnology industry. There are a lot of new nanomaterials that are being used and being produced, and there's been very little attention paid to whether they're safe or not. I think you all know that when you get nanomaterial, its properties are very different than the normal size properties. So nanosilver behaves very differently than, for example, regular silver particles. And I can tell you that there's a lot of concern. Some of the nanofibers are long and skinny and are made with active metals on their surface, and they look like asbestos. And in silk culture, they act like asbestos, and at least in animal studies, they behave like asbestos as well. So I think there's some concern that some of these nanomaterials may, in fact, have a potential to cause adverse health effects. That being said, we are all, every one of us are using, if you're using sunblock today, it's usually made with nano titanium dioxide particles or nanosilver. Women's cosmetics are loaded with nanoparticles and so on, and our understanding of what the potential adverse effects of these still needs to be determined. We're actually have funded in 2010 almost $30 million worth of research on nanomaterials, trying again to develop some predictive power, what are the characteristics of nanomaterials that may or may not cause problems. I think that's where we want to go. And then the last point I wanted to make is the whole issue of climate change. And many people, at least in my country, are finding it very hard to believe that climate change is really happening. They say, how can you talk about global warming when when the Northeast this year and the Midwest and the South actually has had these terrible, terrible snowstorms and ice storms, and they've had the coldest winters on record and so on. They forget that last summer, at least in our country, the same areas that have had the coldest winters had the hottest summers ever on record. And the important thing about climate change is its extremes. It's going to be extreme climate events, more so than even specific hot or cold climate events. But anyway, I think the scientific consensus, despite what some of my political colleagues want to believe, the scientific consensus is that climate change is happening. And we actually led an effort last year across the U.S. federal government to develop a research strategy for what might be the health impacts that we're going to need to deal with climate change. And we actually, in Environmental Health Perspectives, which is a journal that my institute publishes every month, and it's a totally open access online journal that we publish in Chinese and Spanish as well as English. But the point is that we actually went through many disease or organ systems, talked about asthma, respiratory allergies, and airway disease, for example, and what are the impacts likely to be of climate change on these endpoints and how will we study them. I think we all know that when we talk about air pollution, and when people talk about climate change, they usually think about the carbon dioxide and the increased carbon dioxide leading to general warming. Remember, air pollution is also ozone, for example, and PM. And we know that if you just reduce air pollution, never mind the CO2, you immediately get tremendous health benefits. In my country, there are about 170,000 excess deaths a year associated with air pollution. So think of the health benefits that we would get by controlling some of the pathways that lead to climate change. Cancer, people often think about, as the temperature rises, there'll be increase, for example, skin cancer, but other types as well. Clearly, changes in infectious disease paradigms. I often joke about, may take some of my congressmen the realization when we start having malaria in Maine, they'll then believe that in fact, we're actually getting mosquitoes in places with carrying malaria that we didn't have before. But anyhow, this is an interesting document that if you're interested in, what are some of the research needs to understand the health impacts of climate change that you might want to take a look at. And with that, I think I've said a lot, and thank you very much for your attention, and I'm happy to answer questions if we have time. Thank you.